There you go, 6 years ago. Yeah, that's what you can say that to yourself.
We'll start the chapter on blood. We turn the amplifier on. Blood is a liquid tissue. And we describe it as a colloid suspension. Basically what you have are formed elements, which I'll define. Essentially you can consider them the blood cells. They're evenly mixed with plasma. Plasma is the liquid part of blood. It circulates as a viscous liquid in your blood vessels, which you just took a test on. So you can separate these fractions of blood. When you fractionate blood, what they're doing is blood draw, right? Do a light centrifugation, and it just takes a few minutes. Done it myself. You just kind of spin it down. And you have the fractions of blood. Fractions. So if you ever see the term whole blood, it's blood. All the fractions mixed together evenly. But if you were to do a little centrifugation and see the individual subfractions, a little test tube of blood, got your top mark here. You know, the top portion, that yellowish, that's about a little more than half of blood. It's the plasma. That's the liquid part. That's straw colored. Now there are things dissolved in it, and I'll go over what those things are. The plasma, the yellow part, the liquid part, is a little more than half of blood. They say about 55% plasma. It's just a textbook average. Obviously, it varies from individual to individual. Now, there's a very thin white layer. It's about 1% or less than 1% of whole blood. So I'll just kind of like leave a little layer that's white. It's called a buffy coat. The buffy coat contains, well, if it's white, it contains white blood cells. That's easy enough to understand. White blood cells are also called leukocytes. Now, leukocytes make sense when you know that leuko means white. And then, well, also within the buffy coat are the platelets, also known as thrombocytes, because they can initiate blood clotting, which I'll probably get to tomorrow. So all that white layer is the buffy coat. Everything below that, all the red stuff, is the crin. red blood cells are in there, obviously, it makes it red. So call it something like 45%-ish of the whole blood. Hematocrit, or sometimes just call it crit for short, contains your erythrocytes. Because urethro means red. You can just call them your red blood cells, or your white blood cells, or your leukocytes, or your erythrocytes. Well, anyways, those are the subfractions from blood. If you ever donate your blood, they keep them all. There's uses for everything. Usually, when you donate blood, like when I talk about it probably tomorrow, you're donating, this is what you donate, the crit. You donate crit. And you know, there's a catastrophe and people, they need blood. This is what usually people need. The oxygen carrying component of the blood, crit. Donate RBCs. I'm sure they have use for the other fractions. They don't throw away human blood. I mean, you donate, they save everything. And there's a use for it. But you donate the crit, generally speaking, for my lecture, that's what we're going to know. Now, here are some kind of like facts and pictures of real blood. So like I said, blood is a colloid suspension. Whole blood, that's what it looks like, scarlet red. If you let blood sit or spin it down, again, you get your fractions, okay? And so, some stats about blood. Okay, the pH of blood with facts. 7.35, 7.45. There's a range. It's a little basic. But the reason why there's a range is because CO2 acidifies the blood. And there's a little more CO2 content in venous blood. So this is basically the pH of venous blood. This is the pH of arterial blood. It's about 8% your body weight, which is remarkable. I mean, blood is the fluid that circulates that keeps you alive. It's only 8%. Okay. I mean, I've been saying in this class, there's about 5 liters in an adult. There's on average a little more for males because on average, males weigh more. So they have more tissue that needs blood. We have about 5 liters of whole blood that circulates. Okay. Now the interesting thing about whole blood, if you draw it and let it sit in a tube, and let's say you siphon off the plasma. Plasma, among other things, has clotting factors soluble in it. Just blood just sitting in the tube will clot. They clot all by themselves. Normally blood is circulating, and the clotting factors don't have anywhere to accumulate. If they do, you get a clot. Okay. So if you're just sitting in the tube, you accumulate and you form a clot. If you siphon off what's left, that's serum. So serum is plasma without clotting factors. If you don't need the clotting factors as part of your analysis, or wherever, for whatever reason you did the blood draw, just take the serum. If you need the clotting factors, well, you can heparinize the tubes. There are ways you can treat the blood and treat the tube so it doesn't clot. You just keep the whole plasma in there. Okay, well anyways, those are some definitions. Functions of blood. Now, I usually don't teach this because in terms of blood gases, that's pretty much covered in respiratory. I already mentioned pH. We talk about pH regulation towards the end of the course when I get to kidney. I've already talked about this quite a bit. You know how you have superficial veins and arteries don't run with them, that kind of a thing. It helps regulate body temperature. And there's a whole chapter on immunity, so I'll talk about that then. So I'll talk about other things for this next short. But the most important thing, if you take away one thing away from today, that is blood transports oxygen. And blood is oxygenated at the pulmonary capillaries, which you know. Okay. That is the most important function of blood. Transports oxygen. That's a 2, O2. And it's important that you understand that it transports that molecule, O2, as O2. There's no chemistry that has to happen. It doesn't have to, there's not some enzyme that has to work. It just binds the O2 and takes it and drops it off. And so it's a very, very good transporter of that. And it's the RBCs, the red blood cells, because they have the hemoglobin, or HB for short. That's the molecule that can physically bind the O2. We'll get to that. But anyways, you can see that blood is oxygenated in the capillaries of the lungs, and that you just drop off that oxygen at the systemic capillaries. More on that later, but basically you pick it up and drop it off. Okay, great. So that's the most important function of blood. If you get into the subfractions a little deeper, we can talk about a little more what's in the content of blood. First of all, if you consider 100% of your body weight, we're just one big bag of water. Most of the fluids, 92% of the fluids, is not blood. It's fluids in your interstitium, in your cells, outside your cells. But then blood circulates in the blood vessels. That's only about 8% of the fluids in your body. So people always say blood is precious. Now you know why. It keeps you alive, and it's only 8%. It's not very much. So if we break down that 8%, we said already, a little more than half is the liquid part. The rest is formed elements. If you break down the liquid part, well, it's mostly liquid. About 92% of plasma is just water. And I even said in the previous lecture, if you lose blood, how much is a lot? About a liter, right? Because after that, the blood pressure starts to go down. The cardiac output starts to go down because you just don't have enough blood volume to circulate, keep the pressure up in the arteries. All right, well, let's see. So, plasma. About 92% rounding up is good old H2O. Right? However, there are proteins that are soluble in the plasma. For example, they list, all those are pretty important, albumin. Albumin is probably the most plentiful plasma protein, and it provides that blood colloid osmotic pressure that I taught you earlier. It provides osmotic pressure. Remember, that's the pressure that keeps fluid in the blood. Globulins, well, the ones we're going to study, the immunoglobulins. Otherwise known as antibodies. Attack foreign invaders. It's these proteins that circulate. So we also have fibrinogen. Fibrinogen in its soluble form, well, it is a clotting factor. So when it comes out of solution, we call it fiber. Insoluble. And that'll help stop bleeds. Okay, and I'll teach blood clotting probably tomorrow. Okay, so let's kind of just skip down to all the other things. Let's skip this. This is a general list. Let's skip down to the particular kinds of formed elements that are in the blood. Formed elements, basically your blood cells. They got platelets up there. They give you the numbers. Based on the numbers, you can see it's mostly the red blood cells, like about 5 million versus 400,000. I mean, there's mostly red blood cells there. Remember, that buffy coat is only like barely 1% of whole blood, so this is a very small proportion of the formed elements. Well, let's list them anyways. Platelets. Remember, you can also call them thrombocytes. I already said that. White blood cells. And this is where you have different types. You have types that are granular and agranular. I'll just list them as how they've listed them here. They list them in order of prevalence. Neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And so, let's write all those down. Neutrophils. Lymphocytes. We'll spend a lot of time talking about lymphocytes. You ever heard of the term B cell, T cell? That's these guys, lymphocytes. The B and the T's. We'll get into the subpopulations of those in another lecture. The lymphocytes, then you have monocytes. Eosinophils, basophils. Those are all the different white blood cells we'll be talking about. And they always color them purple on pictures like this, even though they're white, because they take a stain, and that's how we see them under the microscope. So those are all the white blood cells and platelets, and well, the red blood cells. I'll talk a lot about the properties of RBCs. Got a lot of slides on those, so I'll just list them for now. In a drop of blood, put it on the slide, students are expected to identify all these different formed elements. And they're called formed elements. They're formed in the red marrow. Formed from the hemocytoblast. Think of this cell as an undifferentiated stem cell. It's in the red marrow. An undifferentiated stem cell. They form in red marrow. And let's remember our anatomy, where red marrow is located. You get all of your blood cells from this tissue. It's inside the bones. Inside your flat bones is cranium. Cranial bones have red marrow in them. Sternum. Iliac bone. Typically, it's the iliac where the oncologist might go to take a bone marrow sample if they suspect some kind of lymphoma or cancer. Proximal femur. These are the locations of red marrow in the adult. Okay, well, let's see, let's move on. So there's a nice picture of a white blood cell, a platelet, and a red blood cell. Our slides look a little different. Obviously, we can't go to 3,500x. I want to first talk about how the red blood cells are formed. There's a process that we like students to know. And this picture can help us. So this is all happening in the red marrow. And again, we see our hemocytoblasts at the beginning. If you're going to write anything down, I would write this, what I added here. If you're going to take a stem cell and commit to become a red blood cell, it requires EPO. Development past the prourethral blast stage requires EPO. EPO. Erythropoietin, I almost just use the abbreviation capital EPO for EPO. And so there's these stages, like for example, prourethroblast. Blast usually means an immature cell that could potentially become a different cell. Pro means before. And so to get past that, you need EPO. It comes from the kidney. If you're hypoxic or something like that, the kidney will churn out EPO. It'll target the bone marrow, and these cells will be stimulated to a committed pathway. And you also need iron and B12. Also, iron and B12 also required for RBC formation. B12 usually allows you to absorb iron from your diet. Well, anyways, when you get onto the developmental pathway, the first phase is ribosome, they call it ribosomes synthesis, phase 1. So let's think about logically what has to happen. If your job as cell is to carry oxygen, you just need to make tons of the protein that does it. How do cells make proteins? You need ribosomes, right, for the translation. And so that makes sense. And now if you're kind of staining slides and you want to figure out which cells are making the ribosomes early on, they call it the urethroblast stage. How do you identify a cell in this stage? Well, it'll present this kind of basophilia. That's the color you see. A nice purplish, bluish, whatever you want to call it, that color. Okay. This guy, this color. Call it basophilia. Cells that have that, they're making ribosomes because you're getting ready to translate the hemoglobin. So think of it as getting ready to make hemoglobin. HB, hemoglobin. So as you go from phase 1 to phase 2, hemoglobin accumulation, identify cells that are in that stage. They call them normoblasts. They present this eosinophilia, stained red. They appear less purple, more reddish. I'll say eosinophilic. Just take a different stain. This is like my first job out of college. They call it an H&E stain, hematoxylin eosinophin. And you just stick the tissue in there with both dyes. And whatever dye the cell picks up, oh, okay, well then it's that cell. So cells that are stained eosinophilic, they stain so because, well, the hemoglobin is picking it up. So you're accumulating hemoglobin. So I'm not going to write that, I already wrote it. Obviously, the cell is accumulating hemoglobin. It's getting ready to enter circulation to transport oxygen. Notice the other thing, you go phase 1 to phase 2, is the cell shrinking or getting bigger as you progress through the phases? Shrinking. Now think about that. These cells circulate. So they have to navigate through the smallest blood vessel, which is what? Capillaries. Right? British people say capillary, and we say capillary. And if you remember, go back and look it up. The diameter of a capillary is like 9 microns. So you got to shrink to less than that to squeeze through capillaries, right? So one thing you do as you accumulate, you start ejecting cell organelles. That you don't need. Okay. Shrink the cell. Eventually, you got to be less than 9 microns in diameter. That's very small. Okay, well, the last phase, phase 3, eject, right? You should eject the nucleus. So at that point, you're done making the hemoglobin. I mean, you can't make the hemoglobin once you eject the DNA, right? So, eject nuke. Done making the hemoglobin, then you're called a reticulocyte. Then you enter the bloodstream. You enter blood through, you remember the leakiest kind of capillary? The sinusoidal capillaries? Yeah, so you enter blood in through the sinusoidal capillaries. So at that point, you're considered a mature red blood cell. And you're done. Red blood cells can circulate for a few months, and then they kind of get old and they die. The spleen is where blood cells go to die. All right, so we see a color change here. Just a different slide showing the same material, basically. From 20 hours, 25 hours through, so it takes a few days from here to here for this process to occur in the red marrow for this mature cell can circulate. Every once in a while you see a reticulocyte. I'm not used to seeing these on blood smears. If you see a lot of them, it could indicate maybe a slow bleeding ulcer or something in your patient, because you see a lot of these and you're thinking, oh, the marrow is making blood a lot because maybe there's a bleed somewhere. Well, anyways, you see a lot of these. The RNA, the ribosomes, they drop off as you start to accumulate your hemoglobin. So the erythrocytes are for transporting the oxygen, and some characteristics of them. They transport CO2 as well as O2. Probably mentioned that. So RBCs, they transport O2 as well as CO2. Well, I should say the hemoglobin does. So if you ever see like, you know, HbO2 versus HbCO2, as we'll see in future lecture, the name for this is oxyhemoglobin. And the name for the other one is carbaminohemoglobin. It's kind of fun to say, carbaminohemoglobin. But anyways, more on that when we get to respiratory. The shape is unique. I have a picture of it in the next slide. It's biconcave. And, well, that kind of flattened shape gives it more surface area compared to just a regular round cell. It ensures a shorter distance to the surface to make the gas exchange at the lung or the tissue to be easier. So here's several pictures, okay? And when we look at a drop of blood under the scope, we don't see plasma stuff, we just see the formed elements, that's what you see, okay? And if you look at an individual cell, they kind of like, light shows through the middle because they're squashed in the middle. This shape is the biconcave shape. The word biconcave is used to describe that shape. Okay, I kind of mentioned that already. All right. Curved on both sides. That's biconcave. Curve in on both sides. So at anywhere inside the cell, you're a short distance for easy diffusion. And I guess remember the average diameter, 7.2 to 8.4 microns in diameter. Because remember I said you got to shrink down to less than a capillary diameter, which is 9. So this is the average range for RBC. They're tiny cells. They're the tiniest, right? The only cells that are tinier are sperm. Everything else is bigger. Like, for example, the biggest cell is the ovulated oocyte, which is like 40,000 times this size. Okay, so these are small cells. Well, to put this in perspective, a hair is 50 microns. You can see a hair, right? So imagine, you know, 8 versus 50. These are super small. And they can squeeze through capillaries, single file. Look at that. And the way they, see how they stack, like a stack of pancakes or something? They call that ability of RBCs to stack and file through single file, like standing in line at Disneyland or something, just go through one at a time. They call that rouleau, the stacking ability. Because they're not beach ball shaped, right? They're flat, and they stack well. Rouleau. I think, rouleau. It's a French word. Stack well, basically. Okay, so know that. Well, here's the structure of the hemoglobin. It's a tetramer. Hemoglobin is a tetramer. What you got are 2 alpha chains, 2 beta chains. That's your tetramer. And each of those subunits has iron-containing heme. And that's the structure that binds the O2. Each subunit contains iron containing heme element. And again, I'll put the binds O2 just to remind you of that. That's what it's for. So each molecule can bind 4 O2s. Well, if you look at the numbers here, if each hemoglobin has 4 hemes and each binds, 1 to 1. So basically, if an RBC has about 280 million molecules of hemoglobin, then one RBC can bind over 1 billion O2 molecules, and that number is a little difficult to deal with. So if you do the units conversion thing, we go through Avogadro's number and all that, and molecular weight and all, basically write this number down. About 1.39 mils of O2 per gram of hemoglobin, that's the oxygen carrying capacity of human hemoglobin. 1.39 mils O2 per gram. Remember doing the units conversion in chemistry? That's one of the first things. Now I tell you, if you don't remember, you should. That is the most important, useful information that will help anyone in any science. The information from that one lecture, I still remember when I was 19. I used it every single day. As a grad student, in the lab, everything I did units conversion. So if you're kind of Scott, I got through chemistry. If you're going to go into the health field, you ain't done with it. You ain't done with it. So just get used to it. Okay, I'm telling you. The units conversion is like the backbone of science, of any lab tech or doctor. Okay? So you get used to the whole units conversion. I didn't do it. I just told you about it. But it's going to come up for you if you enter the field. Just saying. Okay. Oh, that, you remember like the nomenclature test? You had to memorize, like, HCO3, what is it? Oh, that's bicarbonate. That is important too. It's like this is that, the molecular, oh man. You do not escape chemistry. It stays with you. And I hated chemistry. I was just like you guys. I hated it. I took years of chemistry. It's like, why do you keep making me take this stuff? I even went to my advisor and said, do I have to take biochemistry? He said, yes. He was right. In biochemistry, you spend a whole test on this. A whole test. One unit is like the study of human, okay, we'll just keep going on. But for this class, let's move forward. Let's do hematocrit determination is very important. You got a patient and you want to this will be the field of hematology oncology. So hematology is the study of blood, and usually a hematologist, who's a doctor, treats people who are anemic. But the reason why they kind of merge it with oncology, which is cancer, is because, well, there are two main categories of, I don't know much about cancer, the two major categories of cancer are solid tumor and the liquid cancers, which are the blood cancers. And usually, when you get into medicine and you want to treat cancer, you don't mind treating people who are also anemic, so they kind of blend the two. So then you just send them all to the oncologist, hematology oncology, one department, one doctor. And well, one of the things, if you want to help your anemic patient and you want to test for oxygen carrying capacity, measure the crit. Okay, it's a quick way to do it. And so what you do is, I got a little thing here. Okay, normally it should be about 35%, right? Should be. So the normal range, HCT, hematocrit, the normal range is like 37 to 54. That's kind of like the textbook normal range. For you have regular oxygen carrying capacity, the normal range. Okay? So what you notice is there's no units because it's unitless. What matters is the proportion, right? That's a little tube of blood. And got your top mark. Whatever the top mark is, doesn't matter. The proportions matter. So, when you spin the blood down, or I already did this, I'll just kind of briefly color the bottom in red. Again, this proportion, that's you want it to be about 37 to 54% of the whole blood. That's the proportion that you're going for. And so the way you can calculate it is, you have the X over Y. So I guess Y is your 100%. And the crit is the X, that's the part. So when you take a proportion, when you take a percentage, right? Because the other thing I remember from chemistry, I learned the percent, it's always part divided by the whole, right? So what's our whole? The Y, whatever that is, and then the X equals your, that's how you will determine, okay? And obviously you can put numbers in and figure it out. That's a quick way to estimate. So let's say you want to dig a little deeper. You can do other things. Let me define these terms here. You may not know that anemia actually means blood. So if you're less than 37, anemia means low blood. If you're greater than 54, call it polycythemia. Maybe if you live at altitude, like in Denver, where the air is thinner, or in Lake Tahoe, and you acclimatize, and well, you're getting less oxygen per breath, so the body compensates by producing a higher crit, so you have more oxygen carrying capacity. The only thing you worry about is if blood gets too viscous and too gloopy, you can get spontaneous strokes because the clotting factors accumulate. I mean, blood just won't circulate if it's too viscous. Okay, so the other thing you could do is you could kind of take a little drop of blood and you can break the red blood cells and you can directly measure the amount of hemoglobin that the cells are carrying. You could look at the cells under the scope. They might be the normal size, they might be small because the cells aren't producing, the bone marrow didn't produce enough hemoglobin. But anyways, for example, the normal range of hemoglobin concentration per deciliter of blood is 12 to 18. Grams hemoglobin per, I'll just put deciliter. A deciliter is equivalent to 100 ml, right? So that's the normal range. On average, who do you think would have a little more? Male or female?
Male.
Yeah, because you typically have a little more tissue to oxygenate. So on average, so maybe males, it's on the upper end of closer to 18. And maybe for a female, it's on average closer to 12. And what if you have a person who's anemic? You'd expect it to be lower, maybe 9 or 8. You could kind of look up the numbers. Polycythemia, you'd expect to be above 18. And athletes who train tend to have very high hemoglobin. Okay, that is a training effect. Increase the blood. Every time you exercise, you're creating hypoxia in your tissues. That's the whole thing. And then so the body adapts by, this guy's going to keep on working out, which we all should. The body's like, okay, I'm going to make more crit. Well, let's look at anemia. Let's look at the extremes under the microscope. You can kind of see the difference, right? Now that anemia happens to be a form of anemia. I'll get into the types called Sickle-Cell anemia. These are white blood cells, neutrophils, but you can see how packed the RBCs are. Here, what you notice is there's a lot more space between the cells. Cells tend to clump together under the microscope when there's less density. And the shape, the morphology is abnormal. These cells, they kind of pucker because of a genetic mutation. So there are several types of anemia. Remember, anemia means blood, so anemia is low blood. You can have just hemorrhage or blood loss. Obviously, that can cause low blood. Maybe an ulcer, maybe chronic hemorrhoids. Well, I've mentioned hemorrhage before. I mean, the main thing about hemorrhage is we don't want the arterial blood pressure to decline. Could be nutritional. I haven't talked about this before. Nutritional anemia. Some causes could be iron deficient. We have a slide of iron deficient anemia. You may notice something called microcytes. That's basically a term used for, obviously, as the term implies, very, very small cells. RBCs are already small. These are smaller because they're not carrying very much hemoglobin, so they're just very small. Pernicious anemia is a lack of B12, decrease B12. B12 is typically found in meats. It's important for the absorption of iron into the red blood cells. Lack of intrinsic factor. This factor from the gut, well, it helps get the vitamin B12. So it's like you need the B12 to get to the iron, but if you lack intrinsic factor, well, we usually see this as a problem in the elderly. A lot of times their diet is reduced for whatever reason, and they don't intake as much. They just stop eating. So the gut mucosa kind of atrophies, so you lose the intrinsic factor and you lose B12. I took a couple of pictures from our collection. And the iron deficient anemia, you see the clumping of the cells. And also, every once in a while, do you see like a really light looking cell right there? Right there. Those are the microcytes. Normal blood smear looks like that. You can see the difference. Here's basically, again, iron mag, normal versus iron deficient anemia. And yeah, again, you can see the biconcave, there's more flattened as opposed to this cell because of the lower hemoglobin content. I took a picture, no, I took it, it's not as good, but it's good enough. I could point out a microcyte. Okay. Iron deficient anemia, microcytes. Here's a picture I took for the sickle-cell slides that we have. It's a genetic disease, okay? So you have some genetic causes. These are nutritional, basically. Sickle-cell anemia, it describes the morphology part. I mean, not only do you have low crit, the morphology of the cells tends to pucker or sickle due to a mutation that's inherited. The other one that's typically inherited are thalassemias, major or minor. Sickle cells also, carriers can show symptoms. It's minor if you inherit only one faulty gene. It's major if you inherit two faulty genes of the same. Whether it's alpha or beta, doesn't matter. I mean, one gene versus two inherited. Remember, you have the alpha chain and the beta chain. And so if one of those chains is faulty and you only have one good gene from one parent, then the red blood cells are going to be much smaller. So I'm pointing out the sickled or puckered red blood cells on this slide. It's a little blurred, but you can still see the puckeredness. Here's the professional picture of the sickled cell, showing you the specific single amino acid mutation right here at this position. And so if hemoglobin's mutated, it makes the whole cell pucker in its morphology. Okay. Again, here's the other extreme, polycythemia. You have you kind of have normal versus secondary versus severe. So this is just living at altitude, this middle category. You worry about severe polycythemia because of blood circulation problems. It just gets too goopy. And the response for human physiology, for all humans, if you go to altitude, because you can drive up to the mountain, it's called acclimatization. And the physiology is that barometric pressure is decreasing, resulting in less O2 per breath in, inhalation. So to do the math, is basically for each 1,000 feet elevation, maximal oxygen consumption decreases about 3%. Okay? So a good way to experience this, drive up to Lake Tahoe in your jogging attire. And as soon as you get there, get out of the car and go for a run. You'll be sucking air. You're like, oh my gosh, I thought I was in shade. But it really does. The air does get thinner when you're up there. Now, so if you stay, for example, in Lake Tahoe for a week or so, or after a few days, you'll adapt. They call it acclimatization. So here are all the beneficial changes. Okay. Decreased plasma volume, that's a bit of a misnomer. You're not decreasing the plasma volume, you're increasing the crit. So because the proportions matter, it's like that proportion decreased, but you're not losing blood volume. Okay, so look at the carrier capacity. Now you have about 35 grams of hemoglobin per deciliter, so it's about double. Right? You increase the vascularity in the pulmonary capillaries, because that's where you pick up the oxygen. So basically, you're perfusing the lungs better. Perfusion always means blood is flowing through it. Okay, that's kind of what physiologists, the term they use. They call it tissue perfusion, always of blood. You increase the skeletal muscle vascularity for improved oxygen transport. See, these are all beneficial changes. The one thing you can't do is you live here at sea level, go up, train for a few days, and then come back down and compete. You lose the effect. Okay, that's been studied. And you would have to live there like your whole life to maintain these beneficial effects. Okay. All right, so what happens is, there's a nice little flow chart. This shows the importance of erythropoietin. So it looks like the stimulus is hypoxia, which means lack of oxygen for whatever reason. Maybe you drove up to high elevation. Any mountain climbers? No? You may experience hypoxia when you climb mountains, obviously. And what happens is, the kidneys will detect it and kick out EPO into the circulation. It'll target the bone marrow. So they show a femur there, right? And that'll increase the crit. HCT, hematocrit. And that should correct hypoxia. Let's say, for example, you have kidney failure or something. For whatever reason, your body isn't churning out the EPO. You're still exposed to the hypoxia. No correction. The crit will not change. Hypoxia not corrected. So you need the erythropoietin. So one problem that athletes do is it's called blood doping. Autologous blood doping is where a competing athlete trying to get an edge will withdraw their own blood, and you take a few pints, you remove it during your training, right? A few months before your competition. You just put it in the freezer. And then the day before event, you thaw it out, and you just drop a bag of blood on yourself, and just give yourself a little boost. Okay? Because you've been training the whole time, so you should have been increasing your crit all along just from your training, and that little boost gives you the illegal edge. So removal weeks before, right? Train to get back to your whatever baseline, whatever it is, and then give yourself a boost day of, or even during the event. Okay, like they always do the Tour de France during this time of the year. And these guys have to sleep somewhere during the race. You wonder if you walk in their rooms, they got blood all over the place. You know, it's illegal. It's a big problem in cycling, by the way. It's the hugest problem there. It was to the point where you guys know Lance Armstrong, right? He was one of the guys that's he's really bad. He did a lot for testicular cancer, but he was the worst in terms of. I heard he's a kind of a. Well, I don't know. I shouldn't say. It's going on YouTube. I don't know the guy. But there's a report. I read some of the report, and he went out of his way, his whole team, just to kind of dupe officials. He cheated. It's not like he didn't knowingly cheat. He cheated, and he tried. And it's very clear in the report. That's all online if you wanted to read it. But again, the thing you worry about is if you're using your own blood, it's hard to catch you. It's your own blood. What are you going to detect in the test? Maybe if you kind of like took a blood draw here and then a blood draw here, and you're like, wait a minute, how come these values are so different? Even then, it's not a smoking gun. One of the things you could do, it's easy to get caught, is if you use synthetic EPO, because that can be easily detected on a test. So if you knew that, I don't know why you use it, but that's how, I believe that was one of the things Lance Armstrong used. He also took diuretics. A diuretic increases urine output. It'll mask what's in your urine sample. There's a lot of unscrupulous things that athletes will do. Basically, the athlete will know the test. How can I cheat so as not detected? And that's basically what they have to do to compete at a world-class level. It's kind of sad that we push them to that. Anyway, that's a different topic. Let's stop here, and I want to give you the rest of the time to study for your lab practical. But before you do study for your lab practical, let's look at the upcoming week. Again, a few more minutes at a time here.